So now we're bringing together some of our scientific and clinical advisors from genetics to data science to clinical care who have volunteered their time and expertise to help us accelerate research. This panel is moderated by Nathan Guo, who many of you know, leading our science efforts, a fellow ZTTK dad. And this panel is your chance to hear directly from the experts shaping what has been rare disease research progress and what comes next. You've already heard from Dr. Erin earlier, Elli Brimble, Richard just now. I would like to quickly introduce Dr. Maya Chopra, who is with us today, just returning from Cape Cod. So really appreciate you making the time from your trip to be here with us today. Dr. Chopra is a physician scientist at the Boston Children's Hospital and an expert in rare disease, rare neurodevelopmental disorders. She's leading natural history and gene discovery studies that are directly relevant to ZTTK, and she brings deep clinical insights into how we evaluate potential treatments. On the agenda, you also see we were supposed to have Dr. Tim Yu, but unfortunately he was here yesterday with us for the CAB-SAB session, but because of family emergency, he's unable to make it today, and we send his best. And he has been another very dedicated scientific advisor for us, but we wish him and his family the best. And please join me in welcoming our panelists and moderator.
Great. So Ada had, in addition to the last survey data I presented, she had also asked me last second to do just an intro quickly, just to frame the conversation, a little bit of a recap of the SAB-CAB discussions we had yesterday, and that'll hopefully help tee up how all of this connects, right? So on the screen, you'll see we have our scientific advisory board. You should think of the scientific advisory board as people who think about the molecules more at the kind of subcellular level. And then we've also got our clinical advisory board. These are your clinicians that you see. Not all of clinicians do research, but many do as well, and a lot of them are represented on this slide. And then importantly, yesterday, we actually had a parent panel where 6 different ZTTK parents, you can see all the different kids that were described, spoke about some of their experiences. And I think we've talked a lot today about how important those experiences are in informing where the science and the research goes. It was a great discussion yesterday. We actually started with the parent panel, so starting with the Sunshines themselves, then went backwards into looking at what are the clinical symptoms and things like that we see, and then finally to what are the molecules that might work. So maybe just as a really high-level framing, how do we think about prioritizing drug development efforts for ZTTK? There's 2 kind of variables we really care about. The first is potential therapeutic impact. Is it going to work or not? Does it actually address the patient need? You'd be surprised how many drugs have been developed and nobody asked the patient if they actually wanted that thing to be fixed. There's the safety and efficacy, of course. You don't want to give your kids something that's not safe. You want something that's disease modifying, so it actually impacts the disease in some way, not just symptomatic. And then, as Richard alluded to, there may be different groups of Sunshines who may react to one drug and another may react to another drug as well. And then importantly, we care a lot about speed to the clinic. So how long is it going to take to actually reach patients? And are there existing medicines like Richard alluded to, or are there drugs that it's going to take time to progress through the pipeline? And ultimately, we try to balance and make sure we have a portfolio of both near-term and long-term options there. So maybe just a really quick recap. There's 2 categories of drugs that we are thinking about. The first is drug repurposing. That's what Richard just spoke about. Many of these drugs are initially designed for one disease or end up being used for another disease. So maybe a more colorful example would be Viagra. I think a lot of people have heard that it started as a heart drug and then they found another use for it. Many drugs are actually sitting on shelves that could have benefit, but there's no economic incentive for pharma companies to pursue them. often these drugs are off patent, so they don't get any protection to charge more money for these drugs. And that's why when Richard talks about these 40,000 medicines that could be helping patients, nobody's really spending the time to go look and see, can this thing help ZTTK patients? And that's why the work that Richard is doing is helping us identify those. I would say that drug repurposing in general has a very wide range of outcomes, right? There might be some drugs we find that are like, I would give this to my kid tomorrow, it's just a vitamin, to, okay, this is a cancer drug, let's think seriously about whether the risk is worth it at the right dose, et cetera. And drug repurposing will tend to be more addressing the symptoms rather than the root cause, but biology is really complex, so it's hard to tell kind of how these things will all interact at the end of the day. And you can see at the bottom of the screen some of the different organizations we're working with on drug repurposing. Then on the following page are genetic medicines. So some of you may have seen some of these stories in newspapers and other news stories or on TV. So there are a really long list of genetic medicines and different types that do different things, and some work for some genes and some don't work for other genes. So it doesn't really matter the different types. In general, these are medicines that treat the genetic root cause. Some of them create an extra copy of a gene, some of them edit a gene, et cetera. And so on this page, you'll see a few specific examples where there have been big steps, right? Dr. Tim Yu, who's on our scientific advisory board and not here, in 2018, developed an N-of-1 antisense oligonucleotide, it doesn't matter what the name is, for one patient, and was able to get it to her in less than a year. London here is getting a gene therapy. This is actually the first gene therapy for a drug called Zolgensma, which is for spinal muscular atrophy. Spinal muscular atrophy is more common than ZTTK and actually was the most common form of genetic mortality. So with the kids with the most severe form of spinal muscular atrophy would actually pass it before 2 years old. And actually this gene therapy now gives them an extra copy and they're able to live. So really groundbreaking results. And then some of you may have seen recently, there was a really big story about this baby called baby KJ, where they actually used gene editing to try to fix the gene directly. So some really exciting progress, but I think the reality that we'll also hear is we're balancing that with, this is still really new science, there's still a long way to go, and we're still learning more about disease and molecules and biology overall. So hopefully that helps frame some of the discussion, and without further ado, I'll go to the panel now. So, maybe a first question here would be, we've talked a lot today about how important it is to understand the patient experience and the family experience. I guess would be interested if any of you have an example of something you've learned or heard from a family in today or in the data that might change kind of how you would think about developing a drug or doing research in some way. And it doesn't have to be any specific order works.
Yeah. So we are working to many different pathways for therapeutic strategies. One is really direct checking, fixing the gene. The other is more compensation. So I talked to one of the parents today during lunch, and then some sunshine, like her case, does not show much the symptom, but he have some metabolic problem, only gaining weight. Now it's like less fat content. So in that case, probably we would change the nutrition, lifestyle, to really change those, the main problem. So in that case, I think it is equally important to think about the very moderate, mild, low-hanging fruit strategy. Now, if while we still choose work to develop real therapy to fix the root cause. So that's one thing we have been working on, but we are not sure how helpful it will be to the community. And then by communicating with many parents, I think this is also the good strategy. So we'll continue work on that direction. So it's really good feedback for us.
I think something that we talked a little bit about yesterday, and it came up again today when Nathan showed the chart around what families were describing as the top concerns for them or where they would want to see the most improvement. I think some of them felt consistent with assumptions that I'd come into the meeting with, but some of them surprised me, and I think it just reinforces this idea of having a partnership with the community as we're thinking about the drug development life cycle. And so all of your contributions to helping to reinforce that partnership is really valuable, because to a point that Richard and both Nathan have made, one of the reasons why drug development in rare disease may be unsuccessful is that it's not that you don't have an effective therapy, it's that you didn't design your clinical trial well. And so I think we're really lucky to have this forum to be able to think through some of those things to design the most effective clinical trial that we can.
And just to add to that, I think one of the things that your question was alluding to was this family-centered endpoints. So when we talk about clinical trial design, and I speak as a clinical researcher, we need to have endpoints that are measurable and where we can show effect in a reasonable time in order to show that the drug works. So it may well work in a dish or in a cell, and it may well work biologically, but if we can't show that in a clinical trial, we're not going to get the right treatment into patients. And this is why it's so important. We hear from you. Speech and language communication are at the top of that list, and I will feed back to you that is very common with neurodevelopmental disorders. And it's really difficult to measure, to design natural history studies that capture really precisely the progression, the longitudinal course of speech, language, communication, and cognition. So I think it's wonderful that you've already identified, and sure, it's preliminary and you will be expanding that data set, but it's great that you've already identified the importance of what's meaningful to you as Sunshine parents and the Sunshine community.
And just building off of that, then what we do is take that information back into the discovery phase as early as possible. And so what I learned was kind of that there are these different, really broad differences among the Sunshines. And what struck me was really, I think as Erin mentioned, the metabolic piece of it. And so just earlier today, I went and kind of thought back to some of the families that I had spoken with, and of the ones that did participate in the study, I was able to look up where they ranked in the groups, and they're all in one group. Again, it's just anecdotal at this point. We need to do more thorough analysis, but that feedback, I mean, that was 24 hours, not even, right? And then that could inform, as you're saying, the endpoints. Like for those patients, maybe we do want to look at how much they walk or eat and other metabolic aspects. So that was really, really wonderful.
Great, super helpful answers. After this, I think we'll start to open it up to more questions, but I'm going to ask a pretty dense one here, which is, I think for a lot of members of our ZTTK community, this is really the first time they're hearing about the possibility of repurposed drugs or new genetic medicines that might make an impact in their child's life. And earlier today, somebody asked the question around kind of how realistic was it, especially at an older age, et cetera, right? So I guess my question is twofold. First would be, what gives you hope in kind of this emerging drug development world for ultra rare, and what are some of the challenges that you're seeing as well? Just to balance, at the end of the day, we want the community to have hope, but we also don't want you to have false hope either. We want to inform you with the reality and also why we're excited. Now, you don't have to answer with the good one first. You can do the bad one first and then the good one, but I'll leave it up to you guys.
I might start with some challenges. And again, this is speaking to neurodevelopmental disorders. The first is delivery to the brain. So our target tissue is the brain. We either need a molecule that can cross the blood-brain barrier, or we need to deliver, for example, with a lot of genetic therapies, such as an ASO, which you heard about, we need to deliver to the brain, and often that involves injections into the fluid around the spinal cord. So delivery is a big challenge. How do we get to the right organ? But even beyond that, how do we get to the right cell type? we're targeting neurons, but which type of neurons? And this is why it's important to build cellular models, which you're also hearing about from Dr. Ahn. A second challenge is alluding back to your question about time points for reversibility, is what's the critical window? At which point can we reintroduce a healthy copy of the gene or a protein or an RNA that makes the protein, or somehow upregulate the wild type? But what's the time point that we need to do that in order to reverse the phenotype, or to treat some of the symptoms, or to prevent progression? And we don't know the answer to that. But we do have hope. The brain has more plasticity than what we originally thought. And we think from other disorders that there may be an early window that we can treat. This will involve, again, advocacy on your part to get kids diagnosed early, to make people aware of the importance of early exome genome sequencing. And then that's something I think you can advocate as a group and across disorders. And then the third major barrier is alluding to clinical trial design and having the right kinds of endpoints and measures so that you can demonstrate success. As far as hope goes, I mean, you all give us hope. You are the living embodiment of hope in this room. And the biggest, I guess, one take-home for hope, I would say, is collaboration across research, across pharma, with families, and to put all of these brilliant minds together to set the path for this disorder and for families.
I'll talk about bad things first. So a couple of things I can talk about. The first is the real financial support. Very practical things, but it is actually the major issue in our case. So we are in academic research lab, so we are not biotech profit company that sells something and then get gaining. So we are not wholly dependent on funding. So research funding, mainly government, and in this case. But a lot of these policy change, and then also lack of awareness, really put us really tough, rough situation. And then that actually keep us from moving forward. So I have millions of things that I want to do if I have enough resources, funding, manpower, and then I really want to do day and night to accomplish and test everything I'm thinking about. But it's the major roadblock. And then the other things which I sometimes just frustrate me is the lack of awareness and then enough sufficient attention from the public. So a couple of years ago, I submitted my grant, big grant, to get funding, and then I talk about rare disease, ZTTK, this is why we want to study this. And then the first review, I failed, because some of the reviewers said this research support only handful of patients. It doesn't really help a lot of big public population. So we have to support for bigger population. That really frustrated me and then just made me upset. So this lack of awareness and then attention from public, it's still there. I can see it's better and better, but still this is the real situation. So we move to the good side. But I can see more technology development. The AI tool, just the Richard present, it's really fascinating. And then it's very hopeful. And then a lot of other gene editing technology. You heard about this great news. And then a lot of exciting story. So there is a hope, a lot of technology development. So that will give us the more option and open new direction. And then I think the most biggest, brightest hopes are people like you. Leader in this foundation. Without you, and then without the leader in this population, the foundation, no, nothing could happen. So I can see the biggest hope from your faces, and then your passion, and then your involvement. So this is the biggest hope, and then we should be the power of leading this work.
I can also start with the bad before moving to the good. I think something I would add to what the others have said is, in meeting a lot of the families here, there's a pretty significant variability amongst individual sunshines, and so I think that creates challenges in clinical trial design. But another perspective there, which I think it will be interesting to talk through with the foundation on how we start having some of these conversations, is that all of the therapeutic strategies that we've discussed have risks associated with them. They're largely considered experimental, and we have more limited human data on their efficacy and long-term safety. And so I think, each family is going to have a really individualized choice around how do you evaluate that benefit-risk ratio and something that's so new. And so I think that's something that maybe the foundation can help support some of those discussions and providing the information so that everyone feels they can make an informed choice around how do you approach an experimental therapy that may have so much benefit but may also have risk as well. And then I think something that I feel very hopeful and kind of sustains me even in some of the turmoil in this space currently is, I think it's really hard to quantify what any degree of improvement means for quality of life. Even though we might not be talking in the terms of cure, there's no knowing what even a 10% or 20% change may mean for someone's independence or how someone experiences the world around them. And I think that any individual would benefit from even, a 10% improvement to their quality of life. And I think that's what we can anchor towards as our goal. And that's very hopeful to me.
Yeah, so in keeping with the negatives first, I am going to flip that around. As someone insane enough to be running a startup in this market, I see negatives as opportunities and problems to be solved, as opposed to just throw up your hands and say, oh, that's not going to work. Nothing annoys me more than someone saying, well, that's hard. I take that as a personal challenge. So I agree with everything everyone has said, so I'm not going to repeat any of that. But I would just say that all these challenges in terms of awareness, and you guys are already doing it. You're not done, but you're on the right track, and it's an opportunity. And the problem that is an opportunity is all the other rare diseases are basically in the same boat. And so you can basically be heard more the more you do it by being the first or being earlier. So again, I see it as an opportunity. With clinical trial design endpoints, absolutely. I would say it's almost harder than to figure out a therapy is to figure out if it actually works. I think that's almost harder. And so then it's families like yours that are actually helping us do that, right? And you tell us what work means to you. And we encounter all kinds of ranges, even within a given disease, what that means for an individual family. And then you have to decide, do you want to look at what everybody has in common? Or, as I think we're finding out, maybe you have to split it up into two groups. And that actually may make things more efficient and stronger. In terms of on the more purely positive side, I think I shared what we're excited about here. In terms of just within a span of on the order of months, we went from, again, I've never heard of ZTTK, right, to having potential, I want to stress the potential part, potential therapeutic paths that are realistic. If the data support it, there are some very realistic paths, right? And so it doesn't have to take a long time. And where we and all these researchers here and also in other communities like yours, there's a lot of precedent for things potentially being able to move quickly. So it doesn't have to be slow and painful forever.
That's great, and a lot of great themes there. And we'll open it up to questions, but I think just to emphasize a couple points I heard, to really put a fine point around what Dr. An mentioned, when we're talking about government funding, it's really the NIH, the National Institute of Health. The government, it's not to say that there's not government waste, but the government does fund research in things like cancer, but also rare disease that wouldn't be profitable for anyone else to fund. And that's the kind of funding that is kind of under threat now and impacts Dr. On's lab and other researchers as well. And then, I think Richard and others covered, Elli as well, and Dr. Chopra. At the end of the day, each family is going to have to make their own decisions, but for us as a foundation, our goal is to provide options, right? We want the families to be empowered and say, here are some choices I can make, and not just be faced with, there's nothing you can do, go home and love your child, which I think many of us have heard from our geneticists. So we'll open it up to questions and really appreciate all the time from our researchers and also from all of you as families. And hopefully you heard how important that is for informing where we go with research. So we'll open it up for any questions. Just raise your hand and Ada will run over to you.
Great. We have 2 microphones here, me and Julie over there. And maybe when you ask a question, you can introduce yourself quickly, your name and your sunshine or your affiliation.
Thank you to all of you. Thank you for all the parents and getting together. This is really a wonderful opportunity for the insight into this condition. I have a question, perhaps I bear with me with the parents when I ask this a little bit more scientific or technical question, but it's regarding what you presented so far is a lot of genetic data. You classifying patients versus genetic. But can we also look at the proteins and to perhaps actually look at the 2 of them and see how they correlate, or at least pathway-wise? They are talking, maybe that can also help you down the road in terms of when it comes to clinical trials. How do you actually classify the patients that can be in the same group?
Yeah, so maybe I can start, and then Erin, you can give your perspective. Yeah, absolutely. So everything that I showed was based on RNA, right? So it's one way you can look at biological systems generally, patients specifically, with this dynamic perspective, unlike DNA, right? So there's the dynamic versus static piece. Proteins are also dynamic. Metabolites also dynamic. We work with RNA, and we work with all the RNA. So it's like 60,000 different transcripts that we detect. What that does is that we're not detecting the proteins, but the proteins have an effect, which may have another effect, which may have another effect. At some point, it turns into a change on the RNA level again. And so we are able to sometimes directly, sometimes indirectly look at the protein function. In the case of the SON gene, we can look at protein splicing, mRNA splicing in our RNA sequencing data sets. So that's something we haven't done, but the data is there, right? So we can look at the downstream activity. We do not look at protein levels, and I think absolutely could be very informative. I know the foundation is supporting that, and I think that, Erin, that's probably more for you.
Yeah, so I think the RNA level and the protein levels are equally important, because RNA level, you can actually pinpoint exactly direct target. So when you measure RNA splicing, the removal of intron part or different exon stitching, the strategy, then you can pinpoint direct target, and then which can be the more important, more primary target. But then, of course, the protein is very important. So we haven't done any protein comparison experiment. So even with mouse tissue or the cell line, definitely that's something we can do. And then ideally, we wanted to do this with the patient sample, but we started with RNA because the RNA can be isolated from much small amount of sample. The very small, tiny amount of cell from your nasal swab, it is possible to get RNA, but protein, you need large volume of sample from the patient. So that could be the next step if we have now all the resources and then also participation from the patient group.
Hi, I'm Michelle Klingbeil, mother of Sunshine Bradley. This is mainly for Richard initially. You gave us two really good examples of sort of an individualized patients where you could alleviate some of the symptoms. And it seemed like the uniting factor for some of these neurodevelopmental problems are sort of sleep disturbances, something along those lines. So the two compounds that you mentioned, first, are they chemically related so that they are targeting something that has to do with sort of the environment, the neural environment that alleviated some of those symptoms? So that's the first question. Second question is, could we take advantage of some of what you discovered with Rett syndrome and the other syndrome as possible avenues for even that little 10% improvement for our own kids?
Yeah, thanks for those questions. With respect to the first one, so those two programs are completely unrelated, right? So there's no real overlap. Vernistat and Unravel 68, there's no similarity in terms of what they're used for. So the other ones, it's actually an antipsychotic. So it's not even a cancer drug. The other one, it's a cancer drug, but used at very, very low doses. So not at the dose that you'd use to treat cancer. What we've found, so this is how we start, is we find what works, at least partially, sometimes from animals, sometimes from patients, and then we ask, how does it work? I don't really care what that starting structure is necessarily, as long as it points me into something like a target or protein that we do want to then engage. Maybe we need a new molecule to do that. Maybe there's an existing molecule that can do that better that maybe we missed in our screen or we don't have in our database, right? So there's a lot of unknowns, but it's function first. If it doesn't work, I don't want to be looking at it because there's too many options. So it's a way for us to narrow down all the potential complex biology into what could actually fix the problem as opposed to just be neutral or hopefully not worsen anything. In terms of overlaps, that's something that isn't the case in those two examples, I would say, but absolutely. So one theme that I think Nathan alluded to and a few others is rare diseases aren't profitable, right? Just to be very blunt about it, that's true. There's very few patients with any one disease to then warrant spending $1 billion developing something because then this is how you get the gene therapies. They cost a decent amount of money to make, and then you have a patient, 10 patients. It makes it very financially challenging. What we're doing, because we are a for-profit biotech in the end, our strategy, and I didn't address that here at all, is exactly that. How can we unify patient groups? And it may not be diseases, as we're finding out. It may not be all ZTTK patients. But one thing that we did, for example, for Rett syndrome, actually maybe that is relevant here, but is that we found Vernistat hits this mitochondrial target. So that's our new target, never before been drugged. Turns out the metabolite of Vernistat, your liver converts it, which normally is a bad thing. In this case, it's actually a positive thing, into this compound, new molecule, hits this target. Great. So we can't dose that target to a patient without phase 1 trials, all the safety and all that, but we can give Vernistat today at low doses, and your liver turns it into the molecule that you want. But what we found is that that's still Rett syndrome, which is about 18,000 patients in the US, so relatively common in the rare space. But what we were able to show is that other patients could also benefit from the same mechanism. So we're actually about to start running 2 clinical trials. One is in Rett, the other one is in Pitt-Hopkins syndrome. Same drug, same dosing, very similar metrics. Same clinical site, same clinician. Again, that's a way to make it more uniform, more faster for everyone, essentially syndicate that drug development. Both groups could benefit equally from the same molecule, but now both groups don't have to support. They're separate molecules. Even better, we found that 10 times as many patients as Rett syndrome alone, so almost 200,000 patients in the US, could benefit from the same drug. Now, it becomes financially very interesting. And that's what we're doing. We're essentially defragmenting rare as a company. And so my presentation didn't deal with the commercial side at all, but that's what we're doing. And so we have this database right now of 70 different disorders, and we have all this public data, about 1,800 diseases. We are absolutely looking at what are the overlaps. And looking at the drugs that got predicted for each of those 2 groups, there are already active programs that are either in animal models already in our lab or soon to go into the clinic that are coming up. And so ZTTK could piggyback off of some of those. And again, it's not about one drug per disease, because to us, that's a ridiculous concept, as I alluded to earlier. But that subgroup of patients could then piggyback off of another subgroup and so on.
I also want to open up the floor to our virtual participants. If you have any questions, feel free to type it in the chat, and we'll try to get to your questions.
So while we wait for any other questions, I think the other thing I'd add on what you said, Richard, is also that it's not only that some of these features are really common across a lot of these neurodevelopmental diseases, right? There may be some of our ZTTK sunshines who actually look more similar to someone with a totally different disease than they do to other sunshines as well. And so just like, with other medications we take, you sometimes have to try 3 or 4 of them, and some of them don't work and some do. So I think it's really important to know that not only are there potential synergies to share with other diseases, but in a large part, the reason why our foundation has been able to accelerate so much is that all these other diseases have paved the way. So we aren't the only ones working on this. We are standing on the shoulders of giants. This has been decades of research funded by places like the NIH to get to where we are today, and that's why we have so much hope.
My name is Christine, and then my son is Andrew, and he's 25. We were diagnosed 2 years ago. And my question is, in doing surveys, and maybe this could be to the Citizen Health ma'am, or the drugs, you're often surveyed for symptoms and milestones and things like that, but are we at all capturing what things did work for all of us so that we can share that information? Because I haven't seen that yet. And then along those lines, the medications that might have worked, and being able to pull off of that. Like, we just started a new seizure medicine that's actually working for a lot of different ways, and we'd love to be able to share that information with somebody, but it's not something that we take the time to put out there, I guess. But if there's a way to capture all of that, that might save on some of the trials, or in the meantime, give us something that we can use. There's 14 drugs that most of the kids take. There's got to be something that we can share on with each other, especially for sleeping causes. What do we take, or what do we have him take for his cocktail, sleep cocktail, that kind of thing.
Yeah, thank you for the question. It's a really great thought. I think we are trying to do this to a certain extent by being comprehensive in the medications that we're capturing from the records, which includes things like supplements, which sometimes may not be studied as much as medications that are prescribed. I think what does this medication worked means gets a little bit more complicated, and so from the medical records we can look for things that feel concrete, but some of the things that we're thinking based on feedback that we've heard from other communities is that there are some really good nuggets in the Facebook communities. And so we're, with the advent of new AI and LLM technologies, thinking about ways to try to mine some of that data to summarize some of the key findings or trends or patterns that are emerging from some of the natural places that you may be communicating about what you're trialing, and then potentially structuring that into things that might be able to be validated with another study design, for example. But it's a really important point, and I think we're starting to think about it, so I'm sure that other groups are starting to as well.
And I want to emphasize too, like Elli's saying, these are things on our roadmap. Actually, one of the biggest barriers to doing so is really important too, which is that, as Elli mentioned earlier, all research with human patients has to be regulated by an independent review board, IRB, and that's meant to protect patient privacy and other issues related to HIPAA. So it's actually something that's in progress. We are working on those types of things, but we have to put the right infrastructure in place before we start to take that data. But it's absolutely something that is on our roadmap and something we're prioritizing.
I had a question for Dr. Ahn. I'm Brittany Nichols, and this is my husband, Brett Nichols. We have River, our sunshine. He's 7. You had mentioned something when you were talking, and I feel like it was said, but maybe we didn't understand it completely. You were talking about your study and where you're at now. Did you mention being able to be close to a gene therapy to try in human trials, or where are you at with the study altogether?
For gene therapy.
Yeah.
So the roadblock in the difficulty of gene therapy for SON is the first, the size of SON gene is really big compared to other gene. So when we look at the protein size, the SON need more than 2,400 amino acid to make entire protein. So that means the gene is really longer than other protein. Average protein size is 300 to 500 amino acid. So it's more than like 8 times, 9 times, no longer than other one. So it is hard, actually, with current technology, put the entire gene as called like a virus particle, and then put in our cell. So that's one, the difficulty. So we are thinking about different strategy, whether we split this gene into 2 pieces and then deliver these 2 together. And then there is a way to connect these 2 at the protein level. So we are working on this with our SAB members lab. And then the other strategy will be so make a mini version of SON, because we know SON has many different areas with different function. So also, as I showed you in my presentation, some lower species has very simple SON. So that tells us some part of the SON is more important than others. So if we make mini version of SON that contain only very essential part of the SON function, and then deliver this as a single fragment. So these are 2 strategies we are thinking about. But yeah, it's not going to be happening the next day and then in the next month. So it will need a lot of study. So we will first study with mouse model. So that's our plan.
And then maybe just to build on that, you asked about gene therapy specifically, but you might remember on my slide there was a whole long list of them. So those are both gene replacement therapy strategies, but there are others as well. So for example, antisense oligos may be a better fit for ZTTK. The strategy might be, we have 1 good copy, can we turn that copy up, for example?
I think that's what I meant, anything.
Yeah. And so all of them will have different trade-offs on how effective they might be and then how fast they can get to the clinic. So I think in general, we think about drug repurposing as probably the quickest path, and then balancing those with the longer-term kind of genetic medicines. And depending on the results of an experiment, that could accelerate to be a year or 2 away, or the experiment might not work and you go find a different strategy as well. So we're exploring all the options and balancing the kind of near term and long term.
Yeah, so for this direct gene fixing mechanism, so it should depends on whether it is missense mutation, nonsense mutation, or frame shift. We should choose different strategy. So that actually we have several example published recently about, it's called the prime editing, which can fix frame shift, and also the CRISPR-mediated editing, which can be really good strategy for nonsense mutation and missense mutation. So these are still on our list, to-do list. And for antisense oligo, so the antisense oligo is a strategy to produce more RNA from the more correct RNA from DNA. Because when you first make RNA message, you have very first draft containing essential part, non-essential part. So some of those non-essential part removal is there naturally. So it can actually reduce the efficiency of making more correct RNA. So if we, block that part and then make more productive RNA, so just to help reducing non-functional RNA, so make more functional RNA. That's the strategy of antisense oligo, ASO. So it's also on our list. Yeah.
May I add
Thank
one more
you.
point? Yeah. I think one of the great strengths of the foundation and the strategy is recognizing that there are a range of potential therapeutic modalities with varying timelines, but at the same time, the clinical trial readiness has to happen. So there's no waiting for the right therapy, that you've already moved so fast to build a clinical trial readiness strategy, because that's a no-brainer, and that will be needed regardless of which treatment you end up with.
Great. So I actually have two related questions from our virtual participants, and then we can probably take one or two more questions before we have to give you all a break. So the first question is from Kathy, and actually Kathy Sunshine is 44 years old, just diagnosed. I'm really happy that she will be participating in our focus group virtually tomorrow. Thank you, Kathy. And anecdotally, I think she's one of the two sunshines that at least I know from scrolling Facebook posts, one of two in their 40s. So her question is, can participants still join the biosample, still join Unravel sample collection?
So, I mean, as far as I know, I think we still have it open. It's more of the foundation's decision than anything else, but absolutely. And then again, we mail the samples, sample collection kits to wherever the families are. So that's not a problem, but yeah, I believe we're still collecting samples.
Yeah, and it's something the foundation would think, what is the incremental cost versus the incremental benefit as well? So, you know, we have a list today. If 5 more samples changes that list, then it'd be more likely to say, yes, let's get more and more people in. So we'll see what the results say, but obviously continue to think about how we can include more people in the journey.
Great. And the second related question from Natalie is, how can international participants join biosample collection and other data collection efforts?
I can speak to Citizen. So international families can sign up for Citizen. We are limited in collecting their medical records because in the US, we leverage some of the US law that gives patients rights to their own medical records. But if those international families have records of their own, they can upload them to their profile. And what we recommend is just really focus on getting the genetic test report updated, because then we can validate those variants and provide that information back to the foundation, which is really important when we think about prevalence and some of the information that other funding bodies may want to invest more back into the ZTTK community.
And like I mentioned with Unravel, so assuming we are collecting more samples in the future or ongoing, then it's no problem. There's, again, if we can ship via FedEx or some other mailing system to you and get it back, then you can participate. There are some countries where that's not possible, but very few.
And maybe the one other thing I'd add is we've, of course, explored as a foundation many ways to try to get biospecimens from international to the researchers. There are real barriers to doing so that make it really hard, sometimes even to cross state lines in the US. So it is something that we're continuing to work on. Other foundations are working on as well, but it's definitely not the easiest today.
Hi, I'm a grandfather of Miles Sunshine. He's out there, he's the one screaming. Everybody probably can hear. I guess my question is, if a couple has a Sunshine baby and they want to have another one, are the chances higher or lower, or what is the odds?
I think the question was, if a couple has a baby that's a Sunshine, has ZTTK, what are the odds of having another kid with ZTTK?
I can speak a little bit to that generally as a geneticist. So the idea would be that the chance would be low. It would be lower. It would be low, but it would be a little higher than that for the general population. And the reason for that is that there's a theoretical possibility that there's a small cluster of egg-making or sperm-making cells that also carry the mutation or the variant. So we would always recommend for couples who are thinking of having another pregnancy to speak to a genetic counsellor to get personalised advice, to get a full family history, and to take into account their own views and considerations about what that means. I find talking to families about risk I think it's always very interesting, because a 1% risk to some would be seen as, oh, that's pretty low, 1%'s okay, and others would be horrified as 1%, but that could happen. If I had the 1 in 50,000, I could have the 1%. So I do think that it's worth definitely exploring that with a genetic counsellor. I think it can be, it's a giant leap of faith for couples, for many couples that I see. I don't speak for all of them. It's a giant leap of faith to have a second pregnancy and to feel that this very unlikely thing happened, and what if it happens again, or if another unlikely thing happens, and all of a sudden you're aware of all these disorders and you listen to talks and you start to hear about them. So I think it's very natural to have this feeling like you're taking a giant leap of faith. And I hope that you manage to find the right kind of team to support you in that.
I think the other thing I'd add to that, a follow-up question to that question that I get a lot is, what about for my other children? Is there a risk that their children would have ZTTK? And that risk is much closer to the general population.
And to be clear, the risk at the general population level is extremely low, right? Just to be clear. And then maybe two points I'd add to I think, Maya, that was a really great point. I think, I don't know how many of you know this, but one symbol of the rare disease community is the zebra, because clinicians are often told, when you hear hoofbeats, think horses, not zebras. Well, all of us in this room have found a zebra, right? And so we start to think like, okay, that's going to happen every single time. But it is something that is really difficult to, and as Maya mentioned, you should talk to your clinician and really think through together. And then, as some of you may have seen, Ada is expecting in November. And so just to give a little bit more pointed kind of framing, there are options you can consider to evaluate. All of them have trade-offs. None of them is perfect, but there are options to genetically test in utero, to avoid, to check whether somebody has ZTTK. But again, all of this should be done in consultation with your clinicians, and there's trade-offs and benefits for risk benefits for each of the approaches.
Do you know of any families that have 2 children with ZTTK?
I don't think so.
Documented or written up at all?
The only ones are the inherited, maybe missense variants. But actually, from a de novo perspective, I don't think we've, as a foundation, encountered a single family that had 2. And to be clear, like the de novo mutations, someone can correct me if I'm wrong, every human amongst us, every adult here, even non-ZTTK, has maybe 30 to 80 de novo mutations out of billions of letters in the DNA. And most of them do absolutely nothing. And so that is really, the odds of that happening multiple times is staggeringly low. The only exception to that is the one that Maya mentioned, if a cluster of sperm or eggs happen to have that same mutation as well. But again, at the end of the day, all of this should be done in consultation with your geneticist. And yeah.
Oh, thank you. Thank you. I can stop screaming now. And she actually recommended doing a sperm sample because it's obviously non-invasive and a way to look to see if there are clusters, at least in the sperm. And so, I imagine that certain insurances are willing to cover that kind of thing, others are not. But something else to talk to your geneticist about, because I think it's kind of a easy way to get a little bit more comfort, obviously not total comfort. But.
Great, well, thanks everyone. This is the end of a great panel, but certainly not end of the conversations. Let's just give the panelists and our moderator a big round of applause. Thank you.
Thank you so, so, so much.